with such type of bacteria, which is acute bacterial infection, is the neutrophil. As far as you see, the neutrophil nucleus made up of loops. Normally, 3 or 2 to 5 loops. 2 to 5 loops. It is phagocytic cell. The neutrophil is phagocytic cell. Increased number of neutrophil known as neutrophilia. You've neutrophilia here there associated with the acute bacterial infection. Neutrophilia is indication of acute bacterial infection. The second type of granulocyte is the eosinophil, as we mentioned, and the eosinophil is phagocytic cell, but also it is secretory cell. It secretes histamine, and it is increased in the allergic reactions. Any skin rash or any allergic reaction like rhinitis or hay fever will lead to increase in the eosinophil number. Will lead to eosinophilia. And also it is associated with parasitic of intestinal parasites, like a hookworm or a tina, whatever it is, tina saginata or tina solia. Increase or any infection with the parasites at the level of the small intestine will lead to eosinophilia. Eosinophilia, increased number of eosinophils. The basophil, as we mentioned, is not a phagocytic cell, it's a secretory cell. It secretes histamine and heparin. And it plays essential role in the allergic reaction. It plays essential role in the allergic reaction. And all of you know the role of histamine. What is the effect of histamine on the blood vessels? As vasodilators, to increase the blood flow to the site of inflammation. To increase the blood flow to the site of inflammation. Also, it contains heparin, and all of you know the heparin is natural anticoagulant. The heparin is natural anticoagulant. It prevents occurrence of clot at the site of the inflammation, so as to facilitate a continuous blood flow to the site of inflammation. And it is non-phagocytic cell. Non-phagocytic cell. The agranulocytes, actually these cells contain not clear and obvious granules in their cytoplasm. These are the monocyte, lymphocytes and monocytes. Both of them are made up of one nucleus, containing one nucleus. The nucleus is not lobuated. In the lymphocyte, the nucleus is round shape, and in the monocytes is kidney or oval in shape. Kidney shape or oval in shape. The lymphocytes play essential role in the immune response. As far as you know, their role in the immune response is very crucial, and it is highly selective. Highly selective, not like the other phagocytic cell. It contains large purple nucleus, and the size it is a little bit more than greater than the red blood cells. And all of you from the previous session, you know that the diameter of the red blood cells is about 8 micron, and the lymphocytes are larger than the red blood cells. The monocyte is the largest cell in the bloodstream. Its diameter may reach up to 22 micron, and it is highly important phagocytic cell in the body, because it is present in the bloodstream and it migrates to the tissue. Actually produced as well as the other cells from the bone marrow, red bone marrow, and it stays only 1 to 2 days in the bloodstream. Then it transmigrates to the tissue so as to act as important phagocytic cell, which is known as macrophage. Macrophage, it is a Latin Greek name, means big eater, and it plays essential role in the immune response at the level of the tissues, and it is of very long lifespan. It may stay within the tissues for months and even for years. All of you know the red blood cells lifespan is? Pardon? 120 days, yes. And as far as we are going to know about the platelets, they are of very short lifespan of 8 to 12 days, but the white blood cells, of which the macrophage is important, is of very long lifespan, up to years. The lymphocytes, as we mentioned, from morphological side of view, there is two types of lymphocyte, large lymphocyte and small lymphocyte. Both of these are greater, their size is greater than the RBCs. And from functional side of view, there is two types of lymphocyte. All of you are aware enough about these two types, which is the T lymphocyte and B lymphocyte. T lymphocyte, responsible for cellular immunity. It acts against the microorganisms by secreting of certain substances to kill the microorganisms, and the B lymphocyte responsible for humoral immunity. And we mean by humoral immunity a type of immunity in which the cells secrete mediators circulating in the bloodstream, which is the antibodies. And all of you from the previous session also know that the antibodies are plasma proteins. مش كده؟ Belong to which group of plasma proteins? Gamma of gamma globulin. Yes, gamma globulin. Globulin of gamma region produced only by the lymphocytes. Is the only type of plasma proteins which is produced by the, not produced by the liver. All of the plasma proteins produced by the liver cells except the proteins of gamma region, which is the antibodies produced by the lymphocyte. And the lymphocyte, the T lymphocyte of two main types, expressing different types of antigens. And if they ask you about what's the antigen expressed by the T cytotoxic cells, it is CD8 مش كده؟ ما تقول الكلام ده موجود في السلايد يا اخوانا. It's quite clear in this illustration. That means the cytotoxic T cells express CD8 and T helper express CD4 antigen. And the T helper cells act to help the cytotoxic cells, and it helps the B lymphocyte to produce antibodies. Actually, there is huge number of T cells, up to not less than 18 type of T cells. Importantly, there is T suppressors, مش كده? T suppressors and T helpers play an essential role in the pathogenicity of the AIDS, which is the HIV virus. The formation of leukocytes, which is leukopoiesis, formation of RBCs known as erythropoiesis. All of these cells, as we mentioned before, their precursor is hematopoietic stem cell. Presence of a specific mediator lead to differentiation of hematopoietic stem cell to a specific blood cell. What is the mediator which differentiates the hematopoietic stem cell to give us RBCs? Allah يفتح عليك. Erythropoietin is a mediator which is lead to differentiation of hematopoietic stem cell to develop into RBCs. Here we have two mediators which they are acting so as to differentiate the hematopoietic stem cell into white blood cells. These are the colony stimulating factor and interleukins. And both of these two substances produce in the, or their concentration increase in the blood upon exposure of the subject to foreign body, like the microorganisms, whatever it is, parasites or viruses or bacteria. These microorganisms stimulate white blood cells the available white blood cells and other endothelial cells to secrete these two substances, choline-stimulating factor and interleukins. Both of these two mediators play essential role in the activity of the white blood cells. Firstly, they are stimulate the choline-stimulus, the hematopoietic stem cell to be differentiated into white blood cells. And also they are activating the available white blood cells to perform their function, whatever it is to secrete the content or to phagocytic the to engulf the other foreign bodies. These are the two stimulating factors. For this reason, there is no sex difference in the number of white blood cells. As we mentioned in the previous slide, a number of white blood cells normally is around 7,000 cells per cubic millimeter. We didn't mention that of males or females, because both of males and females, if they are exposed to these two mediators or to the bacterial or infections which lead to increases of these two mediators, their white blood cells will increase. In males and in females and males, in regard to the red blood cells, there is differences. And the base of this difference is the need of the tissues, which is higher in the males for the muscles than the females. And all of you, the number of red blood cells is around 5 million. And it is 5.5 in males and about 5 million cells per cubic millimeter in females. And why the number of females red blood cells is less? Because the number or the mass of active tissues in females is less than the mass of active tissues in males. And all of you know the lean body mass of males, lean body mass or body mass without fat, and not body mass of the muscles. The muscles in males, the muscle mass in males is greater than the females. That means our muscles more actively, metabolically active than the adipose tissues. That means we are need for more oxygen and we are need for more transporter for this oxygen. That is the explanation why the number of red blood cells and the blood volume is greater in males than females. Not related to the menstrual cycle, because the menstrual cycle, during the menstrual cycle, we're using only not more than 80 milliliter, and it's compulsory. It's going to be compulsory because any decrease of the blood physiologically like this condition of menstrual cycle will be compensated by increased secretion of erythropoietin from the kidneys, and then the number of red blood cells will be reset back after 7 days, 5 to 7 days after the menstrual cycle, or decrease or exposure of the subject of the female to the starting of the bleeding. And why the blood cells will be reset after 7 days, 5 to 7 days after stimulation of erythropoietin? Yes?
Because this is the generation of the dog.
Allah ta'ala. It's the time taken by the hematopoietic stem cell to develop into reticulocyte in the bone marrow. It takes about 7 days. It takes about 7 days. Very good. It takes about 7 days. يبقى if يعني انتوا ناس الميديسين ناس يعني لو عندك subject suffering from severe anemia، عرفت انه السيفير anemia بتاعته دي nutrition anemia. الموست كومن nutrition anemia هي شنو؟ iron deficiency anemia. هتقوم تعمل له شنو؟ المانجمنت بتاع السبجكت ده هيكون شنو؟ ترانسفيوجن. ليه ما تديو ايرون؟ ها؟ ليش ما تديو ايرون؟ ما انت عرفت انه عنده هنا تديو ترانسفيوجن، ليه؟ انت يو ار رايت تديو ترانسفيوجن، تعمل ترانسفيوجن. لكن واي نوت ايرون؟ It takes 7 days مش كده؟ احنا عارف كده البيشنت بتاعنا. very quick and through management to give him iron, to give him transfusion, and then supplement the subject with iron for the normal formation of blood cells, which it takes not less than 5, in most of the reference 5 to 7 days. فأي acute blood loss رايح بدي فيه شنو؟ transfusion, blood transfusion. This illustration will show you the percentage of different cells in the bloodstream. As far as you see here, the monocyte is the dominant cell. Percentage of the monocyte out of the total number of white blood cells is 60 to 70. When this number is increased to 80 or 85, this is a condition of
Acute neutropenia.
it is a neutropenia, which is associated with acute bacterial infections. The increase of monocytes is associated with chronic bacterial infections. And we did mention that, it is stated here in the monocyte increase associated with chronic bacterial infection. An example of chronic bacterial infection is tuberculosis, مش كده؟ tuberculosis هو السل الرئوي مش كده؟ هو السل whatever it is pulmonary tuberculosis or lymph node tuberculosis ولا intestinal tuberculosis ولا bone or bone tuberculosis. tuberculosis ممكن يعني يكون في chest في lungs ولا في bone marrow ولا في bones generally whatever it is yellow or red bone marrow or in the intestine or the lymph nodes. It is a silent disease. One of the, it's a good example of silent disease. Silent, that means of no clear symptoms for a long period of time. Take a subject infected with tuberculosis, they would have 3 or 4 months without developing clear symptoms. After that, symptoms become very mild symptoms, fever at night, sweating, and loss of weight, gradual loss of weight, and then after it become very aggressive after 6 months. Okay, it needs الشباب اخذوا attendance الشباب it is example of chronic TB, tuberculosis, causative microorganism here, Mycobacterium tuberculosis, Mycobacterium tuberculosis. فبالله يا اخوانا give good care to the other side of illustrations. احنا كنا متفاهمين بين الناس body fluids percentage or volume. clear illustration في اللكشر الاول. الناس كتار ما جاوبوها صح يعني. The gift question. The platelets, as I mentioned, is of true cells because it's a fragment of one of the stages of development from the stem cell. And the fact or the mediator which is differentiate the hematopoietic stem cell into platelets known as thrombopoietin. Erythropoietin for RBC, thrombopoietin for the platelets. And at the steps of differentiation, there is cell known as megakaryocytes. The last stage, fragmentation of the cytoplasm of this megakaryocyte will give us the platelets of a diameter of not more than 4 micron. It is not a true cell as we mentioned. And its number it is about 250,000 per cubic millimeter. And of short lifespan, between 8 to 12 days. That's because it contains no nucleus and no production of enzymes within the cell. And we are in need for when the enzymes present inside the platelets is consumed within this period of time. And no replacement will occur because it contains no nucleus. Importantly, the platelets containing important granules, and these granules containing the mediators which is important for their function, like serotonin, calcium, and a number of enzymes, ADP, and platelet-derived factor. All of these mediators are highly important for the platelets function. you know serotonin. Serotonin is a neurotransmitter and hormone, مش كده? And present in vacuoles or in vesicles within the platelets, and it acts as important factor to stimulate vasoconstriction at the site of injury. Exposure of the collagen will attract the platelets to site of injury, and then platelets release this serotonin. Serotonin constricts the blood vessels so as to decrease the blood flow to the injured part of the blood vessel. This is the first step of the mechanism of hemostasis. All of you will have another term which is homeostasis. مش كده؟ Homeostasis is stabilization of the internal environment. And hemostasis is a mechanism in the blood by which all of the mechanisms, a number of mechanisms in the blood act to decrease the blood loss upon injury. And it will start off three steps, main three steps. The first one is to decrease the blood flow to the site of injury by vasospasm, vasoconstriction. And as I mentioned, one of the important mediators of this vasoconstriction is the serotonin. It decreases the blood flow to the site of injury. Then after, if the blood flow is not stopped by, or the blood dribbling from the injured part is not stopped by vasospasm, the second line against blood loss will be triggered, which is aggregation of the platelets. Aggregation of the platelets to form a platelet block. A block of platelets aggregate at the site of injury so as to act as mechanical protector against dribbling or blood loss. And this we call it primary hemostasis. يعني if they ask you what is the primary hemostasis is quite clear here. مش كده هو شنو؟ An aggregation of platelets or formation of platelet block. يبقى معناه عندنا شنو؟ عندنا secondary hemostasis. Secondary hemostasis هو formation of clot. If the aggregation of the platelets fail to stop the blood loss from the injured part, a third line of defense against blood loss will be stimulated, which is blood coagulation. And simply, the blood coagulation is conversion of a soluble protein, fibrinogen, into fibrin, which is solid mesh. Yes, into fibrin, which is solid mesh. What is the factor which is trigger this third step? The primary coagulation. Primary coagulation in which the platelets aggregate. Aggregation of the platelets leads to secretion of enzymes from which thromboxane enzyme is important. And this enzyme trigger activation of thrombopoietin into thrombin. The thrombin activate formation of fibrin from fibrinogen. And fibrinogen will be activated so as to be converted into fibrin. And main factor which is need to trigger all of these activities who are exposure of collagen. Then upon injury, the collagen will be exposed. Normally, no collagen is in contact with the blood flow. But the area, any part of the blood stream at which the discoagulation of endothelial cells occur and collagen is exposed, this will lead to formation of the clot. A thrombosis which occur in the intravascular part without injury is due to discoagulation of the endothelial cells and exposure of collagen, which is trigger the cascade of reactions lead to formation of platelet block and coagulation. Coagulation is the secondary hemostasis. Secondary hemostasis is the process of blood coagulation. Clear يا شباب؟ Any question يا أحباب؟